Hello folks, hope you are doing well. So welcome to another demonstration video. This is a quick one so that you can just follow it as quick as possible. And this particular demonstration video is on a very basic bioinformatics tool. This particular demonstration caters to the public which are very new to this particular subject of bioinformatics. So we are talking about NCBI. Okay, so I will be demonstrating what are the tools as a beginner that you can learn from NCBI, from the likes of your genomics tool we will be discussing today. So from the likes of your dbSNP and ClinVar. So I'll just be taking two tools so that you can understand it very clear and in a very fast and precise manner. Okay, so without further ado, let's get started. Okay, so first things first, so let's type in NCBI in our search engine. So whatever search engine you are using, basically you will be going on to your search engine and typing your NCBI. Okay, so in capital, small, doesn't matter, you can type it in anyways. Okay, so basically NCBI basically refers to a National Center for Biotechnology Information. Okay, so basically this is a site maintained by US government and it has all kind of information. And basically NCBI is one kind of a primary database. So what is a primary database? Basically primary database has all of the raw data from the wet lab based experiment or your dry lab based experiment. Basically experimental based data is stored over here. Okay, so all of the secondary databases, particularly doing your protein modeling or protein sequencing, those things, okay, so those are secondary databases concerning to a particular job or doing a particular job, and all of those secondary databases are curated from this particular primary ones like NCBI. Okay, so yeah, so basically let's get on to this. So first things first, here you will be getting a toggle button. So here in the drop down menu you can select many databases, because inside this particular primary databases you will be having database NCBI, you will be having multiple of other databases. Okay, so here you can see the URL ncbi.nlm.nih.gov. okay, so NCBI, the full form you already know. Then your NLM stands for your National Library of Medicine, which is part of National Institute of Health. Okay, and the site belongs to US government, so that is why .gov. Okay, so now we were talking about two of the tools in today's demonstration. So first is your dbSNP. So basically SNP are single nucleotide polymorphism, as you know. So in order to identify single nucleotide polymorphism, you can head back to this particular tool that is called as your dbSNP, or in actual terminology it is called as SNP. Okay, so it is pronounced as SNP and written as SNP. So it basically refers to single nucleotide polymorphism, which means poly means many, morphism means repeats. Okay, so single nucleotide change that repeats in the genome multiple times, so that is your single nucleotide polymorphism. Okay, so in order to identify that, you can simply go on to drop down menu and select SNP over here. Okay, and once you select SNP, you will be redirected to dbSNP itself. Okay, so you can search via gene term or like variance term or sequence, any of those things you can use. Let's suppose I am using a gene called as your BRCA1, BRCA1, which causes breast cancer. Okay, so BRCA1 and BRCA2 is most widely known for causing breast cancer. Okay, so let's search with that. Okay, so yeah, so it is already going on. So here you can see here you can see it is already an RSID. So this particular thing is called as your accession number. Okay, so basically accession number would be a particular or what do you say, you can say it is a annotation, it is a way of annotation. Okay, and it is unique. Okay, so like you can find that this particular SNV, that is single nucleotide variance, can be found under this particular RSID. RS means reference SNP, and if the SNP is just submitted and not verified yet, it would be called by the name as SS. Okay, SS 1 2 3 4 whatever be the number, so that is the serial number basically. Okay, so let's go inside this and see what are the things that we can get or extract from here, or what kind of information it provides us. Okay, so basically you can go to all of the different type of SNPs that are there. Okay, so like for this particular gene, which are being reported all across the globe. Okay, different what do you say, your studies would be finding different type of variances with different type of locations. You can find different type of variances. Okay, so let's look at that. So here you can see what is the organism. Organism is human, that is Homo sapien. Okay, this is the chromosome number 17 and then the position. Okay, according to GRCS 38.P14, that means this is the assembly actually. Okay, so which particular assembly it is, like you can have GRCS 37 as well. Okay, that assembly is obviously a bit backdated. Okay, this is the latest one, and accordingly what will change, the location will change. Basically this particular ATGC sequence location number would change. Okay, so in the chromosome. Then what are the allelic changes that you can see? Here you can see G is greater than A, G is greater than C, and G is greater than T. What does it signify? Basically in genomic terms, whatever is written as greater than means that is the reference allele, basically present in the reference genome, whereas these are the alternate alleles. Okay, so G is replaced by A, G is replaced by C, G is replaced by T. Obviously G will remain the same because it is in the reference genome, and if you go on searching for the world population, most of the people would be having in this particular position, they would be having G only. Okay, so you can see what is the frequency of those particular alternate allele changes. Here you can see these are the alternate allele changes frequency. Okay, so you can convert it into percentage also, not a big deal. Okay, so likewise, obviously it is in a probability based value, so that is why it will go like it is counting from 0 to 1, that is why 0.35. Okay, likewise. Okay, so now here you can see according to the population studies, what is the relevance of all of altered gene frequency. Here you can see the total population sample study, and then only European, African, then your other Asian country based studies. Okay, here you can see reference allele will always remain the same. Okay, whereas the alternate allele will change, and what is the alternate allele frequency, that does matter. Here you can see in African American population you can't see the G replaced by T, so the probability is 0 actually. Okay, whereas G getting replaced by A is having a higher probability of 0.23 as compared to T. Comparative analysis I'm telling you. Okay, so likewise here you can see, and based on the different kind of study that has happened, okay, like your 1000 genome, okay, and 1000 genome 30x. Okay, so all different types of studies that are also cited over here. Apart from that, you can also compare it to the reference genome and see what is the location of it. Okay, apart from that, this particular RSID can be known by what are the RSIDs referring to this particular same variance, okay, which are there in all different web servers as well, not only dbSNP. Obviously this particular variance, if it is there, it will be noted in all different kinds of tool as well. Here you can see all different kind of tools. Here you can see, apart from that, many things you can see, variance details are there. Okay, so you can explore this particular database. I just wanted to keep the particular demonstration very short, okay, so that everyone can relate to it and use it very wisely. If you want to choose the assembly, if you want to change the assembly, here you can change. Okay, so GRCS 37 you can align it to, or even NG also you can align it to. Okay, you can also see what are the different types of disease it breaks in, basically clinical significance. It is taken mostly from your ClinVar, so I will also demonstrate ClinVar in this particular video only. Here you can see the ClinVar accession ID, what kind of clinical significance it is having. So it is basically benign. Okay, this particular variance is most probably not causing a very harmful disorder. Okay, so like breast and ovarian cancer it can cause, but at benign stage. Okay, so very less chances of this particular variance that we are talking about is being converted to your breast cancer or ovarian cancer. Okay, so if this was uncertain, so a bit more probability it would be having. If it was pathogenic, so highest probability it would be having to be converted to disordered condition. As you understand, all variances are not going to be converted to diseased condition likewise. Okay, so obviously it would be coding for a particular protein. Okay, if you want to see the protein modeling and all, those videos are also there in the channel. You can have a look at it. Okay, so how to do the modeling or what is the relevance of it. Okay, obviously we are talking about multi-omics, so this is the just the building block, that is your genomics we are talking about over here. Okay, so all the submitted part, history, publication that you can see over here, you can read from this. Okay, so wherever this particular variance has been reported, all those papers you can find from here. Okay, the PubMed ID is given, PMID, PubMed ID is given. Okay, so apart from that, you can also look at this particular thing from your Google Scholar as well. Okay, so likewise you can explore the tool. Okay, so quickly let's get on to the next part of the tool. Okay, so we'll so you can again go back, or basically directly go on to NCBI. Let's suppose we are again heading back to NCBI and going on to all databases. Here instead of SNP, this time around we'll be selecting your ClinVar. Okay, so you can see the ClinVar over here. Okay, in the drop down menu. So where is it? So you can directly type in also, and you will be redirected to that particular page. That is also possible. Apart from that, you can select ClinVar. Here you can see, if I zoom in, ClinVar is being selected. Okay, now let's do it with the same gene, just to negate the like confusion, or likewise. Okay, so just as to remove the confusion, just search by the same gene name. Okay, we can do that. So likewise here you can see the search term, it has come, so it is getting, so this is a gene. Okay, so basically you need to signify that like this particular thing, what do you say, the database would be basically taking into account that this is a gene or a protein or a sequence. Okay, so because ClinVar caters to everything. Okay, so gene, it is also having nucleic acid, also it is having amino acid, also it is having protein, also it is having, likewise. Okay, so likewise if I go on to this, okay, so here I would see what is the clinical relevance of a particular variance. As I have already stated that all of the variances are not going to cause a disorder. Okay, so the same thing, that if I want to look at that this particular gene variance, or like this is a sequence, this particular G is, let's suppose, replaced by T or something like that. Okay, I'm not talking about protein over here, only the genetics we are talking about. So if it is replaced by this, okay, so what is the probability of this particular thing to be causing a disordered condition? Okay, so as I have already stated in the previous example, that if it is having pathogenic, so basically ClinVar divides the data into 5 components. So what are those particular 5 components? Those particular 5 components are benign, like likely benign, then your uncertain, then your likely pathogenic, and pathogenic. Okay, so basically pathogenic is having the highest probability of causing a disorder. Okay, disordered condition. So here you can see what is the identifier, same the particular what do you say, the assembly GRCS 38, based on that we have, or it is also called as a HG 38. Okay, so based on that we have basically assembled. Okay, so based on that particular assembly, it depends. Okay, so why? Because if the assembly changes, assembly is what? Basically how much coverage of the whole genome you are having. So 3.2 billion base pair, how much coverage it is having. Okay, so this is the most accurate as of now. Okay, so much more than this has been discovered, but not yet come into picture over here. Okay, so likewise you can see the cytogenetic location also over here. Okay, so the chromosome 17, and this is the location over here. Okay, what kind of variance it is? Copy number gain. Okay, so as you can see, insertion, deletion, multiple things can happen. So basically this is a copy number gain. Okay, so whereas now you can see the multiple things over here. Apart from that, this particular variance is also like present in all other databases, from like your dbVar, it is present in dbSome is there, dbVar is there, likewise ClinVar is there, ClinGen is there. Okay, so multiple tools are there. OMIM is also a tool for your what do you say, if you want to know that what is the variance. Okay, so or what is the particular gene causing that particular variance. It is disease specific basically, and it caters to all of the disorders which follows Mendelian genetics. Okay, so OMIM, online Mendelian inheritance in men, that is the full form of OMIM. Okay, so we are not catering OMIM in this particular session. Okay, so basically we are not taking OMIM. Okay, so here you can see what is the gene, what is the role of it. Okay, according to which particular thing it is assembled using. Okay, so mostly you'll be finding GRCS 38 and 37. Okay, then see what is the significance of it. Okay, you can see the condition is germline condition. Okay, apart from that, the condition is pathogenic. Okay, so it has been reported as pathogenic according to what is the variance condition. So this is the variance, what do you say, annotation that we can see over here at the top also. So this is also called as your, likewise you had the association ID as like starting with what do you say, like your RSID, no? So RSID in your this thing, dbSNP. Likewise here the accession ID starts with your RCV, whatever be the number later on, so based on the serial number. So that is the accession ID. Basically this is unique to this particular variance. Wherever you click on this particular accession ID, you will be redirected to this particular variance only. Okay, so you can see what is the condition. So it causes a pathogenic basically means it is having the highest probability to cause a disorder. Okay, so this particular variance is having the capability of causing a disorder. What kind of disorder? Here you can see. Okay, so submitted as what kind of condition it can cause. Apart from that, you can also see the publication details over here. Okay, the PubMed ID is also given. If you click on this, you'll be redirected to the paper concerning to this. Okay, you can see this is the paper concerning to the same. Okay, so you can read out this particular paper. Apart from that, what more information it provides, you can see the on your right you will be having the drop down menu. You can directly click on that. Okay, so likewise you can see multiple what are the pathogenic, like you can see from here itself, you can see that this particular variance is also having a copy number gain. So this is basically a overview part, and if you want to dive deep into any of the what do you say, variance, you can do that. Okay, so here you can see mostly all of them are pathogenic. Okay, so the star rating that you can see, that is also given over here. Apart from that, so the gene, this is protein change specific, but if you want to go by single allele also, you can go by this. Okay, so this is a deletion. Okay, I was talking about the insertion and deletion. Let's suppose we are talking about this one. Okay, so this is a deletion kind of a variance, and here you can see this is also pathogenic, causing the condition as breast ovarian cancer, familial, basically running in the family basically. Okay, so familial or genetic in normal terms, local terms that you call it as. Okay, so like the offspring would be having from their parents and likewise. Okay, so running in the family. Okay, this particular condition. And this particular paper also you can cite over here. Okay, so what is the submitter details is over here. Okay, likewise here you can see, so n number of things you can do from here. Okay, so you know about this particular tool. This is a very, very useful tool. Okay, ClinVar as well as your dbSNP, if you want to know about the single nucleotide variance or single nucleotide polymorphism, or in ClinVar you can look at clinical variance is the full form of this particular thing, and the short form of it is ClinVar, and that is the name of the tool, and it is in NCBI. You can directly search using your Google also. Apart from that, in NCBI it is better that you are getting all of the tools in one place. Okay, so that is all from my side as of today. Okay, so if you want to know more on this particular tangent, or if you want to learn some more things on bioinformatics, please follow this particular channel, share with your peer ones. Apart from that, also do subscribe. Okay, so that it encourages us to basically bring up more videos for you, and so that you can learn bioinformatics basics for free. Apart from that, we can also go on to advanced versions. So I have also covered docking and protein modeling in this particular channel before this particular video. I will also give it in the end note, or I can provide the link in the comments. So please do check those out as well. Okay, and so that is all. Support us. Thank you. Bye bye.